welcome everybody to this new appointment of our seminar series. So I'm very happy to introduce Robin Thompson, our speaker today. So Robin did his PhD in Cambridge, right? And is currently associate professor at the Mathematical Institute at the University of Oxford, where he's also leader of the infectious disease modeling group. And has lots of experience in modeling different diseases with different modeling approaches. So I'm very excited to introduce the talk today on mathematical modeling early and late in infectious disease outbreaks. Thank you, Robin.
Great, thanks very much. Thanks, Francesca. Thanks very much for the invitation to come and speak. So as you just heard, I'm going to talk about mathematical modeling at the very beginning of an infectious disease outbreak and also at the very end of an infectious disease outbreak. And also, as you just heard, I lead the infectious disease modeling group in the Maths Institute in Oxford. And the sort of overall goal of the group is to develop mathematical methods and mathematical tools that can be used to guide control interventions during infectious disease outbreaks. These are the current group members. There are 3 postdocs in the group at the moment, 3 PhD students, and then there are 2 members of the wider group who are other PhD students where someone else is their sort of primary supervisor. And as I say, we're interested in developing maths to answer public health questions. So before an outbreak has even started, we're interested in using mathematical methods to answer questions like where are outbreaks most likely to happen? And then linked to that, where should surveillance resources be deployed in order to find outbreaks as quickly as possible? In the early stage of an outbreak, when there are small numbers of cases, we're interested in assessing whether those initial cases are going to lead on to a really large outbreak with lots of cases, or are those early cases going to fade out so that you just have a minor outbreak with very few cases? And then linked to that, which interventions can be brought in order to reduce the risk of getting a really large outbreak with lots of cases? Then in the middle of an outbreak, we're interested in questions like how effective are the control measures that are in place and which interventions should be changed or brought in order to optimally balance the benefits of the interventions, so maybe the public health benefits of the interventions, against the costs. So they might be economic costs or they might be public health costs of having the interventions in place. And then finally, at the end of an outbreak, we're interested in questions like how can interventions be lifted without seeing a resurgence in cases? And questions like, is the epidemic over? So, for example, if we haven't seen a case for the last couple of weeks, we're interested in working out a probability that the outbreak is over, as opposed to the outbreak not being over, in which case we might expect more cases to happen soon after. So, like I say, we're kind of interested in mathematical modelling at these different phases of an infectious disease outbreak. We've got a few different projects in the group at the moment. So one of the projects that we're doing is funded by GSK, and it's all about the development of mathematical models in order to optimize vaccine development and vaccine deployment. We've got a Wellcome funded project on climate change and vector-borne disease. And we're also doing work on avian influenza modelling in Great Britain, and that's in partnership with the UK government's Animal and Plant Health Agency. So what I'm going to talk about today links in with these sort of different phases of an outbreak. So I'll start by starting with the kind of before an outbreak phase and the question of where are outbreaks likely to occur. In that context, I'll talk about vector-borne disease and where vector-borne disease outbreaks might be most likely to happen under a changing climate. Then I'll talk about the initial phase of an infectious disease outbreak when you have small numbers of cases and using models to assess the risks that those early cases go on to become a large outbreak with lots of cases. And then towards the end of the talk, I'll talk about the end of an outbreak and this thing I was talking about a moment ago about assessing when an outbreak is finished. So to start then, the question for the first part of the talk is where are vector-borne disease outbreaks most likely under climate change? This is work done by my postdoc, Will Hart, and also by my now former PhD student, Alex Kay. So the idea of this project is to take climate projections, use those climate projections from a range of different climate models to say something about suitability for vector-borne disease or for vectors, first of all, and then to go on and say something about suitability for vector-borne disease as the kind of second step. I'm going to focus on that first part in this talk. So, in other words, taking climate projections and saying something about suitability for disease vectors. And so, the broad idea is to take simulations of models like this one. So this is simulations of the Community Earth System model, one of the primary climate simulation models used in the US, to then pair that with an ecological model. So in this case, what this particular graph shows is it shows patterns of rainfall and temperature which are suitable for Aedes aegypti mosquitoes to survive. And then to sort of couple those two things, so basically to look at when the climate simulations fall within the ecological niche predicted by the ecological model, to say something about the number of days per year that where the climate is going to be suitable for vectors in a particular location in a particular year. So that's the kind of broad, kind of overall idea of this work, and I'll unpack that a little bit in a moment. One thing that I'd like to point out is that these climate simulations that we used from the Community Earth System model account for two different things. So one thing they account for is climate change, so the fact that there's going to be a kind of warming trend going forwards. But the other thing that they account for is what's called natural climate variability or internal climate variability. And that's the idea that year on year, there are fluctuations in the climate, and that's on top of the kind of overall warming trend of anthropogenic climate change. So, for example, if you look in this figure, so what I've done for kind of three locations here is plotted the annual mean temperatures, and the red is data, and then the grey behind it are simulations of the Community Earth System model. So it's lots of simulations all overlaid on top of each other. But what you can see is that if you look in the real world data, you can see some variability there year on year. So in other words, the climate isn't something, the temperatures in the real world are not something that kind of smoothly increases over time, but rather year on year you have variability. And similarly, if you look then at the grey in your climate simulations, there's also variability. And so individual simulations from the Community Earth System model look reasonably like the real world data. But then if you take the average of the simulations from the Community Earth System model, then what you get is the black line. And the black line, as you can see, is something smooth. So the average over the climate simulations all run under identical conditions. And so I guess what you see here is you see the effect of anthropogenic climate change through the black line, but then you also see some variability year on year about that, which is this effect of natural climate variability. And one of the things we're interested in is understanding the impact of this natural climate variability in addition to climate change on suitability for disease vectors. So to assess that, what we did is we followed an approach that have been used by a number of authors previously, which is to develop an ecological model. And in the ecological model, this is a model of Aedes aegypti. We assume that the vectors are separated according to their life cycle stage. So you have eggs, you have aquatic stage individuals, and then you have adults. And the rates at which vectors kind of pass through these different stages depends on temperature and depends on rainfall. So different parameters separately depend on temperature and rainfall. And what we did is we took lab data and then we fitted responses of these parameters. So for the temperature dependent parameters, we fitted the response to those parameters to temperature using the lab data. So this is one example of one of the fitted parameters. So this is the egg hatching rate as a function of temperature. And similarly to the other temperature dependent parameters, what you see is that essentially you don't get any egg hatching if the temperature is too hot or if the temperature is too cold. But there's a sort of optimal temperature somewhere in between, which is the optimal temperature for egg hatching. And then what my PhD student Alex did is he calculated the equilibria of the ecological model at a range of different rainfall and temperature values. And what's shown in this figure in the bottom right is that inside the black dotted line is the kind of rainfall and temperature combinations where you can get a sort of positive equilibrium number of adult mosquitoes, adult vectors. So within this black dotted line, that sort of gives you the combinations of rainfall and temperature values where the vectors can survive. And there's some uncertainty in exactly what that region is, and that carries through from uncertainty in the fitted parameter responses. Okay, so as I said before, what we do is that we then take climate simulations from the Community Earth System model. We essentially look at the temperature and rainfall in those climate simulations, and we look at whether or not we're within the ecological niche that predicts suitability for the vectors. And that allows us to work out the number of days of suitability for the vectors in each location in any given year. And if we first ignore natural climate variability, so we look at the kind of average results over lots of simulations of the Community Earth System model, what you can see is you can see results a bit like this one. So what's plotted in red here are the locations where there's going to be a substantial increase in the number of days of suitability for vectors as we go on through the 21st century. The regions shown in blue are the regions where by the end of the 21st century, there's expected to be a decrease in the number of days that are suitable for the vectors. And so what you see overall, the kind of striking thing in this figure is that there are locations further north, shown in red, and locations further south, shown in red. And they're locations that are expected to become more suitable for the vectors under climate change. And then something else you can see from the figure is, which I would urge some caution in how you interpret it, is these kind of blue regions. So the blue regions are locations which, according to the model, are predicted to become less suitable for the vectors by the end of the 21st century. And I guess all I would point out is that what's happening in the blue locations is that if you look forwards to the year 2100, those locations are ones that are typically becoming too hot for the vectors to survive. And I guess it's worth pointing out that if the locations are too hot, then there are other problems in those locations in addition to vector-borne disease. So I certainly wouldn't interpret this figure as the blue locations becoming better, but rather they're locations that are going to become very hot. And so I would focus on the red regions and this idea that more northerly and southerly latitudes are expected to become suitable for disease vectors over the course of the 21st century. So that's the first kind of result. And then the second thing that we did is we looked at the effect of this kind of natural climate variability. So what we did is we looked specifically in the year 2100, and then in each pixel of the map, what we did is we looked across each of the climate simulations from the Community Earth System model, and we said in the climate simulations, in that pixel, what is the maximum number of months that are suitable for Aedes aegypti in that pixel? So basically, which simulation corresponds to the most months suitable for Aedes aegypti in 2100? And then we plotted that number of months in the left figure. And then in the right figure, we did sort of the opposites. We looked across all of the Community Earth System model simulations in each pixel. And then we looked for the simulation with the minimum number of months of suitability for Aedes aegypti in 2100. And then we plotted that in the right figure. And so I guess the kind of obvious thing here is that the left figure and the right figure don't look very similar to each other. And what that's suggesting is it's suggesting that natural climate variability is playing a big role in terms of future suitability in any given year, right? So if you look in any given year, there's some possibility that there's substantial suitability for the vectors, but there's also some possibility that conditions are not that suitable for the vectors in any given location. So what my PhD postdoc now, Will Hart, went on to do with this work is he extended it, and he extended it to not just look at natural climate variability or internal climate variability, but also to look at a range of different uncertainty in the future climate. So in particular, as well as this year-on-year variability, which is characterized by the kind of top left figure, there's also uncertainty in which climate model you use to make your projections. So if you make a projection with two different climate models, you get two different answers in how exactly the climate is expected to change going forwards. And he also considered scenario uncertainties. This is the idea that sort of different levels of greenhouse gas emissions and other things will lead to very different future outcomes in terms of the climate. And he took these three different types of uncertainty. And first, he applied a method that's used in the climate science literature for partitioning future temperature projections according to these different uncertainty sources. But then he applied the same method in the context of suitability for vector-borne disease transmission. So this is kind of the novel bit down in the bottom right. And I guess what the bottom right figure is saying, sort of as you would expect, is that by the time you get towards the end of the century, then scenario uncertainty, so precisely which climate scenario you're in, is making a big difference to your projections. But something that is noticeable is that kind of no matter when you look in future, natural climate variability is playing a substantial role in a given year, how uncertain you might be about the number of days that are likely to be suitable for transmission of a range of different vector-borne diseases. So really our message from this work was, well, epidemiological modelers often consider different future climate scenarios. So they account for scenario uncertainty. They also often look at projections under different climate simulation models. So they also consider model uncertainty. But internal variability, the idea that there's kind of natural variability from one year to the next in the climate system, is something that's not often considered in vector-borne disease modeling studies. And so that's something that potentially ought to be considered more in climate-sensitive vector-borne disease modeling studies going forwards. Okay, so to summarize this part of the talk, so before an outbreak happens, models describing climate suitability for vectors can be paired with climate projections to figure out which locations are most likely to see vector-borne disease outbreaks going forwards. As I showed before in the result before considering natural climate variability, the risk of outbreaks of vector-borne diseases like dengue is likely to spread to more northerly latitudes and more southerly latitudes during the 21st century. But perhaps the most important result that I showed in terms of its novelty is that a combination of climate change and this kind of year-on-year natural climate variability are important in determining future vector-borne disease risks. Okay, so that's the first part of the talk. So for the second part, I'm going to change tack completely and no longer talk about vector-borne disease outbreaks, and instead try and address this question. So the question is, when a pathogen first arrives in a host population, are the initial cases going to fade out, or are we expecting to see a large outbreak with large numbers of cases? And it's clear that if you look at outbreaks of a range of different pathogens, you can see either one of these two possibilities happening in historical data. So here, for example, we have the final sizes, so the total number of detected cases in historical Ebola outbreaks. And so if you look, for example, in the year 2000, so Ebola, of course, the virus survives in animal populations, it comes into humans every few years. And when that happens in the year 2000, there was human-to-human transmission, and I think 451 detected cases in the outbreak in that year. But in another outbreak 4 years previously, there wasn't sustained human-to-human transmission, and so there was an outbreak with just a couple of cases. Okay, so when a pathogen comes into human populations, you might see sustained transmission, or you might not. And these two possibilities also come out if you run simulations of stochastic epidemiological models. So if you run a simulation of a stochastic SIR model, for example, you might see an outbreak that looks like this one. If you run another simulation, you might see an outbreak that looks like this one. So, it's a very similar outbreak, but just because of the precise sequence and timing of events, the outbreak looks a little bit different, but nonetheless, it's sort of qualitatively the same. But if you run another simulation, then you might see a minor outbreak like the red one. So in other words, the first case or first few cases might fade out without leading to sustained transmission in the host population. So this leads on kind of naturally to the idea that there's some probability when a pathogen first arrives in a host population that you get a large epidemic, otherwise known as the epidemic risk. So if the epidemic risk is 0, what this means is if you run lots of simulations of your model, all of your simulations will look like the red. If the epidemic risk is 1, what that means is if you run lots of simulations of your model, you'll get big outbreaks like the blue and the black every time. And in reality, the epidemic risk is almost always somewhere between 0 and 1. So in other words, for a fixed set of parameters, there's some probability that you see an outbreak that's big, like the blue and the black, and some probability that you see an outbreak that's small, like the red. And it turns out that you can calculate the probability of a major outbreak, so the probability that those initial cases will lead to sustained transmission. And you can do that in a couple of different ways. So obviously one way is to run model simulations and then just simply count the proportion of large outbreaks. Another possibility is to use a method based on probability generating functions, where you look at the offspring distribution. And another possibility is to do what I'm going to present now, which is to do a kind of first step analysis, as it's called in the paper in the bottom right. And what you do is you say, well, let's suppose that we start with a single infected individual, and then we write down QI to mean the probability of not getting a major epidemic starting from I infected individuals, in which case Q1 is the probability of not getting a major epidemic starting from one infected individual, and then 1 minus Q1 is the probability of a major epidemic starting from one infected individual, which is the quantity that we want to find. And what you can do, so if, for example, the model that you're using is a stochastic SIR model and you're starting with a single infected individual, what you can do is you can basically condition on what happens next, starting from a single infected. So you can say the probability of not getting a major epidemic starting from one infected individual, well, that's either an infection event happens next, and then you've got two infected individuals, or a recovery event happens next, and then you've got no infected individuals. And then, so Q0 is the probability of not getting a major epidemic, starting from no infected individuals. And if there are no infected individuals, there definitely won't be a major epidemic. So Q0 is equal to 1. And then Q2 is the probability of not getting a major epidemic, starting from two infected individuals. And if you've got two infected individuals, in order to not have a major epidemic, what you need is you need your first infected individual to not initiate a chain of transmission that leads to a major epidemic. And that happens with probability Q1. And you need your second individual to not start a chain of transmission that leads to a major epidemic. And that also happens with probability Q1. And therefore, you can say Q2 is approximately Q1 squared. So this is the standard kind of branching process approximation. So you say Q2 is Q1 squared. And then you have this equation to solve. So we have a quadratic equation for Q1. We can then solve that. You get two solutions, which is 1 over the reproduction number of the pathogen, or 1. And then the risk of getting a major epidemic is then just 1 minus Q1, which, so long as the reproduction number is bigger than 1, is given by 1 minus 1 over the reproduction number. And this is a kind of commonly cited value for the probability of a major epidemic starting from a single infected individual. Okay. And calculations like this have been the basis for a range of different publications that we've written in the group for models of various different complexity. But essentially what you get is you get plots like the plot in the bottom right, whereby if you have a more transmissible pathogen with a higher reproduction number, then your risk that any introduced case initiates sustained local transmission is higher than if your pathogen is less transmissible, as one might expect. Okay, so what you can do then, so I guess I talked about a scenario just now that corresponds to a stochastic SIR model, but if you want to, you can extend exactly the same argument that I just presented to you to models with any level of complexity you like. So for example, in the paper in the bottom right, I looked at a situation in which you have two different host types. And this was based on early data coming out of Wuhan at the beginning of the COVID-19 pandemic. And in that early data, it seemed like there were some individuals who immediately on showing symptoms report disease to the authorities very quickly. And there are other individuals who report disease more slowly. And so if you have these two different population groups and you want to work out the probability of a major epidemic, what you do is you can write down the probability of no major epidemic starting from I individuals in the fast reporter group and J individuals in the slow reporter group. You then write down an equation for the probability of not getting a major epidemic starting from one fast reporter. And if you've got one fast reporter, well, then what happens next is either they infect another fast reporter and then you've got two fast reporters and no slow reporters, or the fast reporter infects a slow reporter and so then you've got one of each, or the fast reporter recovers without infecting anyone else, and then you've got no fast reporters and no slow reporters. Then what you can do is do a similar thing for the probability of not getting a major epidemic, starting from a single slow reporter. Then you make this branching process approximation that I just talked about, which is to say that Q20 is Q10 squared, and Q11 is Q10 times Q01, and similar approximations elsewhere. You say the probability of no major epidemic is 1 if you haven't got any infected individuals at all. And so this buys you two simultaneous equations to solve, one for Q10 and one for Q01. You then solve them, and then the probability of a major epidemic, starting from a fast reporter, say, is then 1 minus Q10. So this sort of like shows how you can build some additional complexity into to working out the probability that you get a large epidemic starting from a single introduced case. Another example that we looked at during the COVID-19 pandemic was looking at a scenario in which you had age-structured transmission. So essentially, it was taking calculations of the type that I've just been talking about and embedding a contact matrix in those calculations. So I think most people here are familiar with these contact matrices, but what these contact matrices say is they say the expected number of contacts per day that an individual with age on the x-axis is likely to have with individuals with age on the y-axis. And so kind of notable features here are that this diagonal is very dark, which means that people tend to have contacts with individuals that are the same age as themselves, roughly. And then you also have a grey kind of line up here, which corresponds to children having contacts with their parents or with adults of their parents' age. And there's a line down here which corresponds to sort of parents having contacts with children. And what you can do if you want to work out the probability of a major epidemic is something very similar, but for this for this essentially a model with the number of groups corresponding to the number of different age groups shown in the contact matrix. I think yeah. So what you do basically is you write down Q, I, J, K, and so on to be the probability of not getting a major epidemic, starting from I individuals in age group 1, J individuals in age group 2, K individuals in age group 3, and so on. Then you can write down Q1000 for the probability of not getting a major epidemic, starting from one individual in age group 1. You can then write down Q010000, starting from one individual that's infected in age group 2, and so on and so on and so on. Like before, you then condition, starting from a single infected individual in any specific age group, on what the next event is. You apply the branching process approximation. And then however many age groups there are, what this leads you to is it leads you to that many simultaneous equations to solve for Q1000, Q01000, and so on. And then you solve that, and then you can get results. If you look at 1 minus Q1, for example, Q1000, that will buy you this bar here. If you look at 1 minus Q01000, that will buy you this bar here, and so on. So this gives you, as a function of how old your initial case is, it sort of buys you the probability of a major outbreak. And in the particular analysis I've shown on the screen, I guess unsurprisingly, what you find is that the individuals that have the most contacts, which are individuals in the 15 to 19 age group, are the individuals where if they're the index case, there's the highest probability of getting a major outbreak. And so what Francesca, my then PhD student, did was went on and sort of extended this analysis to consider not just contact dependent transmission, but also scenarios where susceptibility or infectiousness might vary by age too. You can also do similar things for a range of different other types of models. This is an example which maybe I won't go into in any detail, but this is an example where you have sort of seasonality in transmission. In that scenario, what you can do is you can derive an ODE for the probability, so or for 1 minus the probability of a major outbreak, starting from a single infected individual introduced into the population at time t. We've also looked at a few other situations. So this analysis shown on the left was a situation where we were looking at how the probability of a major epidemic is affected by a vaccination campaign. So one of the results that we found in this left paper was in the particular location we were looking at here, even with once you rolled out the vaccine, it looked like you didn't completely eliminate the risk of getting large outbreaks. And then something else we've been looking at is we've been looking at how you can include within host dynamics in the kinds of calculations that I've been showing you up until now. And that might be important if you want to implement a control strategy to reduce the probability of a major epidemic, where the control strategy affects the within host dynamics. Okay, so that's the second part of the talk. So in that part of the talk, we looked at the initial phase of an outbreak and estimating the probability that initial cases lead on to a large epidemic as opposed to simply fading out. I talked about how stochastic compartmental models can be used to estimate that probability. You can generate estimates of the probability of a major epidemic analytically, and I talked about one method for doing that. You can inform those estimates using outbreak data, and then you can adjust those estimates in real time. And I also talked about how estimates of the probability of a major epidemic can be extended to include a range of different features. So there was a model in which there were two different groups. So this kind of heterogeneity in reporting model. I talked about this age structured model, and I also talked very briefly about how you can include temporal heterogeneity like seasonality in some of these calculations. So then for the last part of the talk, I'm going to move on to this question of when can outbreaks be declared over, and can we use epidemiological models to try and infer when an outbreak has finished? End of outbreak declarations are clearly a beneficial thing if they're done well. Um, Declaring an outbreak over allows interventions to be relaxed and some interventions to be removed, but you don't want to declare an outbreak over if, as soon as you remove your interventions, then you immediately see more cases and potentially a resurgence in transmission. And so the criterion that WHO has for Ebola outbreaks is they wait until it looks like an Ebola outbreak has finished, and then they wait for a period of 42 days without any cases. And if there's a period of 42 days without any cases, then they say that the epidemic is over. So they then declare the outbreak finished. So this was what happened in Liberia during the 2014 to 16 Ebola epidemic. And what you can see from this graphic here is that they declared the outbreak over in total 4 times. And on the first 3 times, there were additional cases in Liberia. So it was only on the fourth occasion that there was then no more Ebola transmission in Liberia. And that's not to say that the WHO declarations were wrong. There may have been very good reasons why these additional cases happened. Maybe the virus was imported from other countries, but rather this was motivation for us to start thinking about whether mathematical models can be used to determine when outbreaks have finished. So the question that we wanted to answer is, how long is it necessary to wait without seeing cases before you declare an outbreak over? So in our initial analysis of that question, what we did is we developed a stochastic SEIR model. And in the SEIR model, we differentiated between two types of infectious individuals. There were individuals in this I compartment who were individuals that are infectious and report disease to health authorities. But there were also individuals in this C compartment, these sort of cryptic infectious individuals, who are individuals who are infectious, but they don't report disease to the health authorities. And what we did is we basically ran lots of stochastic simulations of this model. We waited until we'd seen a period of 42 days, let's say, without a case in the I compartment, the reporting disease compartment. And then we basically looked in the E compartment and the C compartment to see whether there are individuals still there, in which case perhaps the outbreak is not actually over. And we did this over lots and lots of simulated outbreaks. And we calculated the proportion of those simulated outbreaks where, after 42 days without cases, the outbreak really was over, in which E and C were both 0. And what you can see here is, with a 42-day period, we found that you can be about, for the baseline model parameterization we used here, you can be about 80% sure that your outbreak's over after 42 days. And if you want to be more certain than that the outbreak's over, then you can instead wait for longer. So if you wait for about 60 days without cases, then you can be about 95% sure that the outbreak isn't over. We also looked at other possibilities. So another possibility is to sort of keep this 42-day rule, but instead do something to increase the proportion of cases that you find. So if you go and do active surveillance and you find more cases, then again, you can be more confident that the outbreak is over after any fixed time period without seeing cases. So that was one of the recommendations from this paper here, was that you need to do active case finding in order to increase the confidence that an outbreak is over after some defined period without cases. So what this does is it provides a sort of general rule based on how long it's been since you last saw a case as to how likely the outbreak's over. But actually, this is all based on a premise that waiting a fixed time period since you last saw a case to then declare your outbreak over is a good idea. And in fact, this may not necessarily be the best thing to do. And just to kind of demonstrate that, well, imagine, for example, that you saw the disease incidence time series shown by the green bars, and suppose that the true end of outbreak probability based on that time series is given by this black line here, right? So as a function of how long it's been since you last saw a case, your confidence the outbreak over is gradually growing. But suppose instead that you saw on this very final day, instead of having one case, imagine that you'd seen 20 cases, let's say. Well, if there were 20 cases on that final day, then you might expect that the end of outbreak probability would then be smaller. And that's because if there were lots of cases on that final day, that's indicative that more transmissions might happen in the time period soon after that. So in other words, if there are lots of cases on that final day, you'd expect a lower end of outbreak probability. And if there's just one case on this final day, then you might expect a higher end of outbreak probability. And if you based when you declare your outbreak over on a fixed time period since you last saw a case, you're not accounting for example, how many cases there were the last time you saw cases in the outbreak. And so what this got us thinking is, well, we might want instead to account for specific disease incidence time series when we calculate the end of outbreak probability. Diseases with very different time series might have a different end of outbreak probability if there are then 42 days without cases. So the idea then was to take a specific disease incidence time series and then try and calculate the probability based on what you've seen up until that day that you're not going to get any more cases in future. So we weren't the first group to think of doing this. So a group in Japan led by Hiroshi Nishiura derived an approximate formula for the end of outbreak probability under a branching process transmission model. So I won't go into too much detail about how this model works, but essentially for every case, you kind of sample, if you want to run a simulation of the model, you sample from the offspring distribution to work out how many cases arose from any specific case. And then you sample multiple times from the serial interval distribution to work out when those secondary cases happened relative to the primary case. Okay. So you have a very basic kind of branching process transmission model. And what we did is we came along and showed that if you have one additional piece of information compared to the information assumed by the Nishiura model, then you can exactly calculate the end of outbreak probability under that branching process transmission model. So specifically what we did is we said, well, what we showed is that if you have information about who has infected who in that outbreak so far, so if you have a transmission tree for your outbreak, then under that transmission model, you can exactly calculate the probability that you don't see any cases in future after today. So the problem with this, I mean, in some ways this is good because I guess this means that you can exactly calculate the end of outbreak probability. And indeed, we showed that you get different answers if you use the exact calculation compared to if you use the approximate formula that Nishiura and others came up with. But of course, the benefit of the Nishiura method or the Nishiura formula for the end of outbreak probability is that you don't need to have the transmission tree, whereas our method assumes that you have the transmission tree in order to be able to calculate the end of outbreak probability exactly. So what we then went on to do is we went on to develop a kind of follow-up to this initial paper, where we then said, well, if you don't know the transmission tree, what you can do is you can work out the end of outbreak probability, the probability of no future cases, by sort of enumerating over all of the possible transmission trees, where this term here, the probability of transmission tree I, is the kind of probability that that particular transmission tree is the transmission tree given the disease incidence time series. And of course, doing this takes a really long time because the number of possible transmission trees for any disease incidence time series is really huge. So what we did instead of enumerating overall possible transmission trees is we estimate the possible transmission trees using MCMC. So this gives us a list of kind of equally plausible transmission trees to then scan over. And then we can use the formula, the exact formula for the end of outbreak probability for each one of those inferred transmission trees, and then work out the end of outbreak probability that way. Um, and so what this kind of shows up till now then, so as I said, WHO declares Ebola outbreaks over after 42 case-free days. Simple rules of thumb for end of outbreak declarations can then be tested as we did using model simulations. But actually, what you might want to do instead of having a very kind of simple rule, like a kind of fixed time period since you last saw a case before you declare an outbreak over, what you might want to do is declare an outbreak over based specifically on what you've seen in the outbreak so far, so based on the specific disease incidence time series. And so this group in Japan, led by Hiroshi Nishura, derived a very useful approximation to the end of outbreak probability. We showed that you can calculate the end of outbreak probability exactly if you know the transmission tree. But if the transmission tree isn't known, then we also showed how you can then essentially estimate the transmission tree and then work out the end of outbreak probability based on your kind of Bayesian estimate of the transmission tree. Okay, so in the last few minutes, I'll just talk about a kind of extension that we then did. So we started calculating the end of outbreak probability based on a specific disease incidence time series using this branching process model, because that was the model that was assumed by the Japanese research group. But what we then started thinking about is whether there's a model which would allow us to do things more simply. And it turns out that if you use renewal equation models instead, then you can write down an expression for the end of outbreak probability much more straightforwardly. So maybe I won't go into detail about exactly what a renewal equation model is, but renewal equation models underlie quite a few epidemiological modeling analyses, including estimates of the time-dependent reproduction number. And essentially what you can do with a renewal equation model, if you want to work out the end of outbreak probability, is you can write down directly the probability of getting no cases every day from day T onwards. And you could do this, for example, for a Poisson renewal model, so a model in which every day the number of cases is drawn from a Poisson distribution. You can do it with a negative binomial renewal model too, so a model in which every day you draw the number of cases you see on that day from a negative binomial distribution. And the reason you might want to do that is that that is one way to account for super spreading. So the idea that on lots of days you might not see many cases, but then on some days you might see a very large number of cases. And we applied this idea in the context of an outbreak of Ebola in 2017 in the Democratic Republic of the Congo. What we did in that paper is we basically looked at the fact that there was an Ebola response team that was deployed. So that was deployed by the World Health Organization and by the DRC Ministry of Health. And what we calculated every day is we calculated the probability of additional cases happening if the Ebola response team was withdrawn and transmission conditions went back to those before the response team was deployed. And so you can see sort of every day your risk of withdrawing the response team, which is essentially 1 minus the end of outbreak probability, gradually declines, such that at the time that the response team was actually withdrawn, you can see that there was a relatively small risk of any further cases happening. And indeed, there were no more cases after that date. Something we did in another analysis is we looked at what happens if you then include underreporting, and I guess it has the effect that you might expect, which is that when there's underreporting, you have to wait longer after the apparent end of an outbreak to then be confident that you aren't going to see any more cases in future. So just to summarize this end bit of the talk, so renewal equation models provide a framework for estimating the end of outbreak probability in quite a straightforward way. So you can essentially just simply write down an expression for the end of outbreak probability. You can inform your estimates of the end of outbreak probability from the data you've seen in the outbreak up until the current time. And as additional data arise, you can then adjust your end of outbreak probability. One possibility, so I didn't talk about this in much detail, but one possibility is that you do you essentially do this analysis every day. So you work out the end of outbreak probability every day, and then you only declare an outbreak over when the end of outbreak probability that you've estimated falls below some threshold value. And that threshold can be set according to policymakers' appetite for risk. Um, Super spreading events can be included in these models, and one way to do that is to use a negative binomial distributed number of cases every day. And then effective disease surveillance is something that's very important if you want to declare outbreaks over quickly and accurately following the last case. To summarize, then, I guess there were three parts to this talk. So, infectious disease models can be used to assess where and when outbreaks are most likely. I talked about that in the context of changing risks of vector-borne diseases under climate change. Infectious disease models can be used to infer the risk that initial cases of disease in a host population are going to lead on to a big epidemic. And infectious disease models can be used to try and figure out when outbreaks have finished. So, I'm going to stop there, say thank you very much. Thanks for the opportunity to come and present. Thanks.
Thank you, Robin.